Director of Business Development US here at IDDI. I'd like to welcome you to our 5th webinar, this one entitled Operational Aspects of Data Monitoring Committees. We are happy to welcome 125 attendees from 12 countries. Before we begin though, just a quick overview of IDDI. IDDI has been providing biostatistical and e-clinical services in phase 1 through 4 clinical trials since 1991. These services, you can see listed on the left-hand side of this slide, include trial design, biomarker validation, data management, statistical analysis including data monitoring committee support, medical writing, and any necessary support for interactions with the FDA and EMA. To support these services, we have developed our own eClinical suite, interactive web response for patient randomization and treatment allocation, EDC, Marvin, and internet coding of medical terms. Now today's webinar will explore the best practices that have emerged over the past 23 years. A review of the safety objectives for the Data Monitoring Committee will be presented. Dr. Herson will also discuss the practices and issues that vary across the pharmaceutical landscape, including some real-life examples. He will then conclude by looking at some of the emerging issues. Now, Dr. Herson will also attempt to answer questions at the end of the webinar, time permitting. So if you have a question, please write it in the chat panel to the right of your screen, and it will be sent to us. Also, at the completion of today's webinar, please take the time to provide us some feedback by filling out the survey form. Now, you can also ask further questions on this survey form as well. Now, these responses to these questions and others that are not answered during the webinar will be sent to all of the attendees in a follow-up email. So who is our speaker today? Well, it is a privilege to introduce Dr. Jay Herson, Senior Associate of Biostatistics at Johns Hopkins University in Baltimore, Maryland. Dr. Herson, a collaborator of IDDI, will draw on his 23 years of experience with data monitoring committees, including the chairing and organizing of the first data monitoring committee in the pharmaceutical industry. And he is the author of the book, Data and Safety Monitoring Committees in Clinical Trials, published by Chapman and Hall in 2009. Dr. Herson, please.
Well, thank you, Paul. It's good to be here, and on behalf of IDDI. IDDI is very much involved in working with data monitoring committees in the pharmaceutical industry in ways that I'll describe later. Go now to the next slide. Here we go. What are the responsibilities of the DMC? First, patient protection. That is to do a periodic review of data in order to see that patient safety is being protected, and to do that independent of the sponsor. Next, if there was one word to describe the responsibilities of a DMC, the word would be stewardship. Stewardship means having the ultimate responsibility for a very important function. Think of a board of directors in a corporation. They have the ultimate responsibility to protect the rights of the shareholders. The officers carry out the different operations, but ultimately report to a board of directors. So we might think, a little slow today, the sponsor as an officer of a clinical trial, and the DMC as a board of directors of a clinical trial, each with their own responsibilities but with a definite reporting relationship. The last bullet I have here is independence. The DMC can only carry out these responsibilities if they do so independent of the sponsor. Working with the sponsor, but nevertheless independently. There are various challenges to independence, and we'll review some of them today. The first known data monitoring committee fully funded by the pharmaceutical industry was in 1988. The company was SmithKline and French. The product was cimetidine, used for stress ulcers. Cimetidine had the brand name Tagamet, but this was, and that was for regular ulcers, but this indication was the more serious one of stress ulcers, where and cimetidine was given IV, not in tablet form in this trial, for patients coming into an emergency room, often in a coma. It was placebo-controlled, and it was to prevent upper GI bleeding. So I was asked to chair this DMC. Of course, we had our safety responsibilities, but also and this is an unusual responsibility for DMC, efficacy adjudication, because there were various signs of the patient. Did the patient bleed? Did they not bleed? By different clinical signs, and someone independent of the sponsor and blinded needed to make that judgment. So that was the first one, and what has been established then in 23 years of data monitoring committees in the pharma industry? Well, people will tell you there needs to be a charter. We must prevent conflict of interest. The committee should have open and closed sessions. There should be a masking or a blinding policy, and someone should take the minutes. Well, all of that is nice, but I had the motivation. Internet is going slowly today. Let me go back now. I'm sorry, I pressed that too fast. Well, anyway, I had the motivation to write this book because it was time to consider best practices. Industry trials are different from government-sponsored trials. And most books and papers that were written by academics had related to the government-sponsored trials. No issues were strictly statistical or strictly scientific. They were all no issues were strictly clinical or statistical. They were all scientific, is what I mean to say. And the DMCs have to make decisions on safety without any knowledge of efficacy, because efficacy is not established till the end of the trial. There's both an art and science in doing that, and that is why I've written the book, and that is what that art and science has developed over the last 23 years. Now try to go to the next slide. Okay, so as an overview today, the emphasis is on safety data. First we'll have an introduction, and then go into safety monitoring, statistical methods, and the biases and pitfalls we will want to avoid, and DMC decisions, and emerging issues. So first, by way of introduction, efficacy. First, why is it that we're putting the emphasis on safety? Because first of all, efficacy is only considered at specific times in a protocol by the DMC, and even then, only when the protocol allows for interim analysis. At the end of the trial, the DMC will be informed of efficacy, but it isn't really their responsibility because they're informed the same time the sponsor is when everyone is unmasked. But safety is always the DMC's responsibility. 85% of the data we collect are for safety. 90% of DMC operations are safety, even when the DMC has some efficacy responsibilities. And efficacy is more subjective than efficacy. And safety decisions are made, again, in the absence of knowledge of efficacy, making the risk-benefit thinking difficult throughout. So we'll see how this is done. It will be important to understand some pharmaceutical demographics. The main distinction I want to make here is there is not one pharmaceutical industry. There are, in fact, three. It's Big Pharma, Middle Pharma, and Infant Pharma. And we can make this trichotomy on the basis of annual revenues, as you see in the slide. But the important thing is Infant Pharma is a startup company. They don't have any revenues from product sales. They have no products on the market. They may be public or they have venture capital financing, but either way, there's tremendous pressures on them. They have to show progress in efficacy and safety to increase their financing or be able to license the product to a larger company. So the vulnerability to negative trial information is considerable for infant pharma and less for big pharma and middle pharma that already have products on the market. Also, the larger companies have DMC procedures in place. Infant pharma does not, and very often they're developed as this trial develops, and in fact, they develop with input from the DMC members, making the operations much different than in an infant pharma company than in big pharma. And we'll see a lot of examples of that as we go through the slides. So who are the members of the safety monitoring team? Certainly the sponsor, the data monitoring committee, the data analysis center. This is a key group, so let me go through that. The data analysis center is that group that will prepare the reports for the data monitoring committee. That is, they will do whatever statistical analysis, whatever listings and graphs are requested. It is best that this be a separate organization from the sponsor. It should not be the same organization that will do the statistical analysis for the new drug application. However, that isn't always the case. Often in big pharma, they will have another group separate from the group working on this clinical trial with a firewall between them. And that is okay, providing it's a sturdy firewall and there are definite SOPs to prevent sharing of information. The IRB and the ethics committee are certainly a part of the safety monitoring team, and the pharmacovigilance unit of the sponsor. This is a group in big pharma. The pharmacovigilance group is the group that has responsibility for constantly monitoring the pre-market safety data as it comes in, independent of the group in the sponsor that's running the clinical trial, even though they're doing it too, and also considerable post-market safety monitoring responsibilities. The pharmacovigilance group is the group that the DMC will report to if they have a safety concern. Now, everybody thinks that if there's a safety concern, the DMC says, okay, we're shutting this trial down. That doesn't happen so fast. It happens after some discussion with the sponsor people who know more about the drug than the DMC people do. And it is the pharmacovigilance group, separate from the group within the sponsor that is working on the trial, that this discussion will take place with. Now, obviously, infant pharma has no such unit, and it is necessary for them to get some kind of consultant independent of the sponsor to have these discussions with. And that is an issue that has not been completely resolved. So the members of the DMC, generally 2 physicians and a biostatistician. One physician may be an AE expert. For instance, in an oncology trial, if the sponsor expects cardiovascular issues as adverse events, they may have a cardiologist as an actual member of the DMC. And then other people, ad hoc consultants, perhaps an allergist or a hematologist, some of those are retained only when we see the only when those types of adverse events are seen. And then there is the non-voting independent statistician from the DAC. This is a person who works for the DAC, who will sit with the DMC members, and this is a person who can unmask the group if they're already still masked as to treatment groups. This is the person who has prepared the analysis and who can even do further analyses right there in the room with the DMC in the closed session if that's required, or at least can take down the requirements for further analyses and do it later. Now, what about the meetings? There is first an orientation meeting, and then periodic safety data review meetings as the trial progresses. Usually the orientation meeting is face-to-face, and some of the periodic safety review meetings might be face-to-face. Many of them are by telephone. Here is an example that might be decided. The first safety review meeting after 50 patients complete 1 cycle, or 4 months, whichever comes first. I put the 50 and the 4 in green because these are just examples of numbers that might be there. If we were to say that the first safety review meeting is to take place after 50 patients complete 1 cycle, well then enrollment might be slow. And if it is, it could take 1 year before that requirement is met. During that time, there are patients nevertheless on the trial who are at risk, and the committee should not wait 1 year just because that threshold has not been reached. So that's why we say after 50 patients complete 1 cycle, or 4 months, whichever comes first. At the orientation meeting, there are various agreements to be made and discussions at least started. I won't go through all of these, but in reading it you get the idea. What is the SAE data flow, the serious adverse event data flow? How does it get to the members? Does it go just to the chair first? Does it go to all the members simultaneously? Is the internet used? Who prepares the minutes, of course, and things like that? What statistical methods are likely to be used in the trial? What would be the format of tables, listings, and graphs that the DMC wants? Will internet software be used to update the DMC members? Different things like this. What is the quality control and software validation to be used? Will the DMC have the responsibility to review manuscripts before publication, package insert, the integrated summary of safety, and other things of that nature? Here is a question. I am the biostatistical member of a DMC working on a trial for an infant pharma sponsor. We've been appalled by the extremely poor quality of data that we're receiving. The CRO is performing poorly in monitoring, et cetera, et cetera. Can we all just resign from this mess? Well, this brings up a question. The answer is no. They have to work with the sponsor, give an ultimatum to the sponsor that things have just got to get better because patients are at risk. But DMC members cannot just resign because it's not a good situation. Because patients are at risk. Just like a board of directors of a corporation cannot all resign because there was a bad earnings report, or because the officers are not doing a good job, because the shareholders are still at risk. So once they join, they must stay with it and work with the sponsor to get things in order when things seem to go wrong. I was asked to serve on a DMC for an infant pharma company. The medical director told us at our first meeting that we can be unmasked at any time. The venture capitalists agreed with them. Should I serve on this DMC? Well, here's a case where a person hasn't agreed to serve yet, and this is unacceptable. The people at infant pharma or any pharma cannot be unmasked. That's the idea of a clinical trial. And the potential DMC member should try to persuade them to remain masked and tell them that the trial will have no credibility otherwise. So there are different masking policies. Partially masked would mean that the members only know A and B. The treatments identified only as A and B. Unmasked, A and B are decoded. And the members are often unmasked from the start because otherwise the members are always guessing which is the control group, which adverse events are coming from the control group, which ones are coming from the experimental group by their knowledge of these drugs, and making decisions based on their certainty as to what these treatments are, and what if they guessed wrong. If they guess wrong, they will be making incorrect decisions, holding meetings of shorter length than necessary, etc., throughout the trial. So it's better to be unmasked from the start. Here's a biostatistician who was asked 7 months ago about serving on a DMC for a phase 3 trial that was about to begin. He called the sponsor several times to reaffirm interest. Each time I was told that the DMC organization was to begin shortly. The sponsor finally sent me a contract yesterday, but also learned that the trial began 2 months ago, and dose adjustments have already been made due to early adverse events. Should I join this DMC? Well, the person has a right not to at this point. The issue here is that what sponsors do not generally appoint physician members to the DMC until they know who all of the investigators will be, because an investigator cannot also serve on the DMC. So while they're still recruiting investigators, they don't appoint a DMC. Nevertheless, there should be a fail-safe date where they say, look, the trial is about to start. We've got to get our DMC in order. These particular physicians will serve on the DMC, and they will not be investigators. I am a DMC chair. The sponsor just sent me and each of the members an email indicating that they are postponing our data review meeting scheduled for 3 weeks from now. They claim the DAC has not had time to do the programming to prepare for the meeting. We don't want to postpone the meeting because it took a long time to get the calendars in order for this meeting. Do we have to comply? Well, first of all, it should be known that the sponsor does not set the date of meetings. Only the chair sets the date. The DMC, through the chair, set the date for their meetings, not the sponsor. So the sponsor does not have the unilateral right to make this postponement. Also, the DMC schedules their meetings in accordance with what they know about safety. And a postponement might put further patients at risk. So this is a bad practice. Right now, the DMC's back is to the wall. They must accept this because the DAC is not ready. And they will have to tell the DAC that this performance is unacceptable, and all meetings must take place as scheduled. Here is a person who agreed to serve an infant pharma company's pancreatic cancer trial, but the sponsor told us that this DMC will meet only once at the end of the trial, because the board of directors is concerned that if we recommend early trial termination due to safety, they'd have to include this information in a press release, would have a bad effect on the stock price. Well, that may be true, but the financial concerns of the company are of no concern to the DMC, whose concern are the patients and their safety. So that in this case, also, they should the person should tell this infant pharma company that this is not a DMC at all. The DMC has to meet during the trial, or they're not protecting patient safety. What is the use of meeting at the end of the trial when everybody has been unmasked? So, safety monitoring. The DMC safety objectives are to protect patients and find dose-limiting or trial termination signals, separate signal from noise, and separate adverse events from the disease process. Those are the things that are going to take place as the DMC deliberates the safety data that they get at their meetings. So what kind of data? Well, here are what I call useful data displays. Certainly, we want to see the enrollment by center, cumulative patient enrollment by month to see that the trial's enrollment is on track and to make suggestions if it's not. The distribution of patient exposure to drug in each group. Some of these are only seen in closed session because they're by treatment group. Reason for discontinuation is certainly important. Some indication of how current the data are. And of course, the treatment emergent adverse events by body system, possibly subclassified by grade, relatedness to study drug, and event types within body system. And this item in 6 again for the serious adverse events, laboratory values of interest too. Now, what are the desirable characteristics for a data analysis center? I won't go through all of these items, but the issue is it should not be a group that knows how to program in SAS or S+. They have to have experience in doing this. They should have SOPs for their DAC operations, software validation SPs. Certainly their computers should be backed up. They should have a validated software already done for this type of activity. Off-site computer backup, as I said, and a statistical staff with knowledge of the clinical trial statistical methods that are likely to come up. So if a DMC member asks for a conditional power calculation, they would know how to do that. The methods that might be discussed with the DAC prior to the start of the clinical trial are on this list. And again, they might want to see an incidence rate and a rate per 100 patient years of exposure, or both. Kaplan-Meier, time to event graph for selected AE types, not for every AE. Landmark estimate for all AE types or selected AE types coming from the Kaplan-Meier. Of course, chi-square tests, a log rank test for the Kaplan-Meier, the odds ratio, or the Poisson rate ratio, or both. I'll explain those later. Confidence intervals, etc. But the DAC should be prepared to do conditional power and some of these others, although that they don't come up very often. I might add that IDDI has a lot of capabilities in working with data monitoring committees and a lot of experience and a lot of good statistical support. Here is a person who is the biostatistician member of a DMC, and been working with the DAC for 6 months now, and this person, the DMC statistical member, is spending a lot of time working with the independent statistician from the DAC, because this person is more of a computer programmer than a biostatistician. And this is not the responsibility of the DMC biostatistician to do that. This is just another case of an inadequate DAC. Here is someone by a statistician member of a DMC, and the person asked for odds ratios and confidence intervals to be computed for the treatment comparisons of adverse events. And after several meetings, this biostatistician member has still not received it. Should I just be satisfied with what I'm getting, or should I continue to press for what I think would help me and the DMC? Well, the answer is yes, you must press for it, because this is important, as important perhaps to the biostatistical member as an electrocardiogram would be to the physician members. I have been in this situation too, and often I think when I don't get the odds ratios from the DAC after at least the first meeting, I always wonder, is it because they forgot, or is it because they hope that I forgot that I ever asked and they wouldn't have to do it? Another kind of unfortunate situation with a DAC. Here is a person who is a chair. The members have been concerned that the sponsor is sending us voluminous tables to review. There are so many pages that present AEs in different ways and with maximum granularity with preferred terms. We are concerned that this information overload might mask true safety concerns. It has the effect of making meetings very long, members getting restless, and a situation where they can't see the trees because the forest is in the way. This often happens when the sponsor says, well, let's just give the DMC all of the tables that are going to be produced for the new drug application, which are, of course, voluminous. That way we'll save money. Well, when that happens, that is not of use to the DMC. And this is why it's so important at the orientation meeting or shortly after the orientation meeting for the DMC members to ask for the types of tables that they need. There are limitations, of course, of a safety assessment by a DMC. They're only going to find AEs that occur immediately and with highest frequency. The DMC is not likely to find rare or delayed effects. They won't develop an understanding of chronic effects, and these patients, of course, are not representative of those who will get the drug when the product is marketed. They will be on there are probably limitations in eligibility and concomitant meds for the trial. And the DMC may miss subtle signals that involve extensive analysis and additional data on surrogates of AEs. So here is a clinical program for diabetes drug, and I just want to show you how sensitive a trial is to detect at least one adverse event of a certain type. We have a rule. The rule is 3,000 divided by N, where N is the number of patients exposed to the drug in the clinical program. So if N is 3,000, 3,000 divided by 3,000 is 1. That means a trial with 3,000 patients exposed is sensitive enough to detect at least one AE that occurs at an incidence of 1 per 1,000. So if there were 9,000 patients that is exposed, there'd be a sensitivity to detect those that would occur at a rate of 0.33 per 1,000. The more sensitive. So here, as we move through this diabetic drug trial, we see that at the end of the first trial with 348 patients exposed, the trial could detect AEs that occur at a rate of 8.6 per thousand, and you see that number get progressively as the cumulative total increases, and at the end of the trial, 2,796 patients exposed, at the end of the program rather, then they would be able to detect AEs occurring at a rate of 1.1 per thousand. Some safety definitions. There has to be a dictionary, which is a hierarchy of event types within body system. MEDRA and CTC are very useful. MEDRA is the ICH-approved dictionary. CTC is used in the oncology trials, and investigators there are used to using CTC. Sometimes they use MEDRA, but software is available for making the translation between CTC and MEDRA. Grading is the degree of severity. WHO and CTC have useful grading scales. AE is the abbreviation I've been using for adverse event, and SAE for serious adverse event. And what is an AE anyway? It's a treatment emergent, and it's an unfavorable or unintended consequence of therapy. An SAE is one that results in death, is life-threatening, or requires hospitalization. There are a few other requirements there, but this is the essence of an SAE. I'm a physician and chair of a DMC, a rheumatology indication. We've been asked by the sponsor for more information from some investigators on SAEs that we have been reviewing. The information has not arrived. How long should we wait before we give up? Well, the answer, as you can tell, is not ever to give up. It takes time for the data to come in from the field, and when it's patient narratives, the narratives often have to be translated from the source language into English, but nevertheless, the DMC has to indicate their expectations on this. I'm a chair of a DMC, a cardiovascular indication. They've requested ad hoc reports to interpret a certain safety concern. The DAC is saying they need several weeks for each table because of the need for validation and the cleaning of the data. And the DMC wants these reports more rapidly than that. Yes, there can be an overrule for validation and quality control. The DAC has to understand that the data won't be completely cleaned until the trial is over. And as far as validation, it would be nice if these were validated before, but if they're ad hoc reports, they just have to do the best they can. There has to be some kind of agreement as to what is a reasonable time, because the DMC knows what that safety concern is, and they want to be able to reach a decision in an ad hoc meeting. Multinational trials present a tremendous challenge, and as all of you know, more and more we have people, subjects from maybe as many as 50 or 70 countries participating in a trial. Of course, the DMC has to consider geographic genetic variation that may result in safety issues in one region but not in other, differential diets. Propensity to self-report AEs. In certain countries, people may be very stoic and they don't want to tell people, especially strangers, about some safety issues they may be having with the drug. In other countries, they may be just suspicious of a capitalist system or something like that. Political, the last one on this page, is in some countries, their health systems require that, in some countries, their health systems give financial incentives to hospitalize patients. That is, if the patient is hospitalized, the investigators are compensated at a higher rate than they are if the patient is only seen in the clinic. So with this incentive, many patients coming in from in these countries with a certain adverse event will be hospitalized where they would not be hospitalized in other countries. Hospitalization is the definition of a SAE. So what happens? We're seeing a lot of SAEs from some countries of a certain adverse event and not others. What the DMC has to do here is look further into these SAEs, patient narratives, things like that, get further information. They can also request that that they will change in these countries these SAEs to AEs only for the use of the DMC and their deliberations, not for anything else, in order to get a common definition across regions. This takes work to do this, of course. And then when that is done, the DAC will prepare the tables according to DMC classifications as well as the original classifications. Now, unmasking in some countries is required when an SAE occurs. So there will be SAEs, unmasking in some countries and not others. But this doesn't matter because the DMC has been, should be unmasked from the start anyway. So it isn't a tremendous problem, but sponsor unmasking is an issue there. And that is something that needs to be handled within the sponsor. There's different medical surgical practices in different countries. Supportive care, how we treat these adverse events, may be different in different countries, and that may affect the severity. Different surgical techniques may be used. In an adjuvant trial for cancer, surgery is not part of the trial. The surgery occurs first, but if different countries are using different surgical techniques or they're trained differently to do the same surgical technique, this may make a difference in the adverse events or their severity. The active control may vary over countries because the same active control drug has not been approved in all countries. And if we're comparing the experimental drug with the active control, we may see different odds ratios only because there's a different active control. Again, this is an important issue in trials. So this has to be dealt with even when the same active control, let's say sorafenib, is an active control and it is approved in all countries, but in some countries that drug is too expensive for the health system to afford, so the investigators from those countries have little experience with this drug, and they may be over-reporting adverse events. And again, when the control group is standard of care, that too may vary among countries. And one thing about the active control, a DMC may see adverse events that are serious in the active control group, and a question that often comes up, can a DMC terminate a trial because of adverse events in the active control group? And the answer is yes, because the DMC is there to protect patients regardless of which treatment group they are in. Now here is a regional representation. Here's a trial where they appointed a Canadian site, started it up, and then the Canadians said we will only participate if a Canadian member is added to the DMC, and pretty soon there'll be Eastern European sites and South American sites, and they'll all be asking for the same, perhaps. So what do we do about this? We can't have a huge DMC, but we can retain different people in these regions as ad hoc consultants to the committee, and once those regions reach a threshold, a number of patients enrolled in the trial, then they, at that point, that a person can become a full member. Statistical methods. The common methods, the incidence rate, chi-square test and p-value, the odds ratio, and the 95% confidence interval. I like the rate per 100 patient years of exposure. And the Poisson rate ratio would be the ratio of the rate per 100 patient years in the experimental group to the control group. The reason for that is there may be different patient years in the two groups, because in one group, patients may go off therapy sooner because they die or because of toxicity. Kaplan-Meier, of course, takes exposure into account. And we can do the full Kaplan-Meier or just the landmark estimate. By the landmark estimate, I mean what is the SAE rate at 12 months estimated from the Kaplan-Meier graph. And the log rank test is the way to test the full Kaplan-Meier, the difference in the curves between time to event between the experimental group and the control group. Now, one hears a lot about multiplicity. I'm not recommending this paper, but it was a very good one in safety data, because we might be testing so many different adverse events and the p-value doesn't really mean as much. On the other hand, in efficacy, of course, if we are going to if we are going to be testing efficacy for one principal efficacy endpoint and 7 or 8 secondary endpoints, that becomes an issue in efficacy. But all we would look for is a p-value or an odds ratio in safety. It's a signal to look further. And clearly, statistical significance does not govern anyway. If we had 4 cases of renal toxicity in the experimental group and 0 in the control group, the DMC physicians don't care whether that's statistically or significant or not. They're going to take some kind of action because that's a very serious adverse event. So I would not worry about multiplicity in these trials for safety. Now, the support graph is something you may not have seen, although some of you may have seen me talk about it before. This is the likelihood support graph, and you see what the graph is. It's standardized to have a maximum at 1. So the most likely value, in this case, this is the event rate per 100 patient years of thrombotic events in the Raffa-Coxev trial. And you see that the maximum likelihood estimator is 1.5 per 100 patient years that you see here. Now, as we come down the curve, the parabolic shaped curve, you see the values just to the left or just to the right are almost as well supported. And the further we come down, the less supported they are. And if we drew the horizontal line at 1/8, for reasons why 1/8 is chosen, I can't go into that now, but you would see a maximum at 1 point. You would see the interval, 1.1, 2, and those are plausible values for this adverse event. So ratio. So the likelihood methods follow the likelihood principle. They do not follow frequentist methods, which depend on repeated sampling, and they're not affected by multiplicity. And the probability of a false conclusion is bounded. So this is really something that people should be looking for use in safety monitoring with DMCs especially. And likelihood methods in the history of ophthalmology. IDDI was a data analysis center for the first VEGF for age-related macular degeneration. And likelihood methods discovered a high incidence of endophthalmitis in the experimental group, and we took action on that and corrected it. We like to think that likelihood methods discovered this differential earlier than would have a frequentist methods, and IDDI is very knowledgeable about using likelihood methods. Well, there are many biases and pitfalls. One of them is granularity. I can best describe it with this table. Within the system order class I disorders, there are 30 preferred terms. 9 of them in this particular trial did not have 0 in both groups. But you see, we get a statistically significant difference there with between treatment A and treatment B, 4 versus 17 events. But one ophthalmologist says, well, this is ridiculous. There's so many 1s and 0s and 2s here. I can't make heads or tails out of this. I want to group the adverse event by the type of inflammation involved. So this ophthalmologist would see that the 4th group that combines 5 different types having a p-value of 0.011. The others not significant. That's certain information that this person has. Another one says, no, I want to group by the type of tissue involved. So there are 4 groups here too, but only the 3rd group reaches statistical significance. I don't see a reason not to do this both ways. Each ophthalmologist is getting some information that he or she needs to give advice. Now, keep in mind that sufficient granularity can keep SAEs off of the label for this trial because I won't find any that differ because there's so many 0s and 1s. And congestive heart failure can be reported any of 3 categories at least that I've listed here. Now, then they're spread out all over. Acute stroke can be classified under several preferred terms, under several organ systems, so that can be spread out. So there needs to be a way the DMC needs to look at this, the way the tables are coming at them, and suggest groupings so that when you get to label time, this will not happen. Obviously, investigator knowledge of the treatment assignment, they may tend to over-report for the experimental and under-report for an active control because they say, oh my goodness, do I have to fill out another form that says there's alopecia with this particular drug? Everyone knows that. I'm not going to do it. But when there's over-reporting for the experimental, under-reporting for the active control, that's when you get differences. Obviously, incomplete follow-up would be another problem. Here is competing risks is something that should always be thought of in looking at safety data. Here is an important example. We have here both treatment A and the other treatment is combined therapy A plus B. Both treatments have 150 patients. However, treatment A has 175 patient years of follow-up, and the combined therapy only 85. And so if we just looked at the cardiac SAEs only by incidence, we get a statistically significant difference showing that treatment A is not as good, and maybe treatment B has a protective effect against cardiac SAEs. However, if we look at the cardiac SAEs per 100 patient years, we don't get a difference at all. That's because in taking exposure into account, the Poisson distribution is taking the extra variability accounted for by less follow-up in the combined therapy. And indeed, in the combined therapy, there may be fewer patient years because patients are exiting this drug due to toxicity or they're dying because of poor efficacy. Now, again, efficacy is a decision at interim analysis when specified, but it's a very simple perfunctory decision at that point. And safety is continuous in the absence of knowledge of efficacy. So what can they do? They can unmask if they see a concern, send a Dear Investigator letter to watch for this, perhaps handle them differently, modification of informed consent, and protocol modification, and trial termination. But that trial termination is the last. Protocol modification would be changing eligibility requirements, doses, and stuff like that that might be contributing to the SAE. Here is a person who's serving on a DMC for seasonal allergic rhinitis, and this person is concerned because there's an 18% incidence of transient headache, which we can confidently attribute to the experimental drug because the active control does not cause this nor any other drug on the market. So this person is saying, well, no doctor in his right mind would use a drug that causes a headache if the other drugs don't. And can they stop the trial because of this? And the answer is no, because the DMCs do not give marketing advice, only safety advice. And this is not an SAE. So at the end of the trial, they may want to contribute that kind of advice. Okay. Here's a person where the accrual is very slow, and this is a DMC member. Can we terminate the trial on the basis that patients are being exposed to a drug where we're not likely ever to learn about its efficacy? I wouldn't terminate the trial immediately. However, the DMC would probably say, look, you need to enroll more centers, you have to do this or that, different ideas of increasing enrollment, and give some kind of a deadline. But indeed, if the enrollment doesn't increase by some kind of a deadline after the advice is given, then indeed they will have to terminate the trial. In adaptive designs, I would just say this, that the adaptation methods at the interim analysis may drop a dose if there are several doses of the experimental drug. But this dose might be the only dose that the DMC is happy with, because the higher doses have toxicity. The adaptation is made only on the basis of efficacy. Again, there'll be a white space, a time for deliberations before the dose is actually dropped, but this is something to discuss with the Pharmacovigilance Committee at the sponsor. Same for most of these. I just go, they're very similar. Same for a seamless transition, phase 2 to phase 3. If they're not happy with the toxicity in phase 2, why should they go to phase 3 merely on the basis of efficacy? And changing the effect size of interest. The CHW method of sample size re-estimation does this. It takes whatever the odds, let's say the odds ratio is at the for efficacy at the interim analysis, and whatever that is, it will increase the sample size so that that odds ratio will likely to be shown statistically significant at the end of the trial. So the statistical member of the DMC must explain to the physician members, look, let's say the odds ratio that we thought was clinically significant, that everyone was agreed was clinically significant at the beginning of the trial was 3. The odds ratio we're seeing now at the interim is only 1.5. Do we want to increase the sample size so an odds ratio of 1.5 will be statistically significant at the end? It's less than the clinical significance, the clinical significance that we thought at the beginning of the trial. And if we were happy with the safety profile with an odds ratio of 3, can we live with it with an odds ratio of 1.5? Important decision, and many people are not aware of this. There are emerging issues, among them, how can new DMC members be trained, perhaps by webinars like this one and my book. Cost control, let me just go over that one. DMCs are making decisions, oh, we need this table, we need that table, we need this one, that one, all of this stuff as the trial progresses. All the sponsor knows is the sponsor gets a bill for this, doesn't know what this is all about, and do they have to pay for it? Well, the answer is yes, but the best way to handle this is for the chair of the DMC to get a budget, and for the DAC to tell the chair, okay, if we make this report, it's going to cost this much, and for the chair, therefore, to be prudent in the spending of the money so things don't get out of hand. Audits will happen now of DMC, so it's important that the records are stored and the meetings take place when they're supposed to, and the charter is followed. Mergers and licensing. When two companies merge or one acquires another, what happens to the DMC? Well, they're still responsible for the safety of the patients. Unfortunately, the people at the sponsor are not so concerned about the trial very often because they're looking for new jobs and they're wondering what's going to happen to me in this merger. Same thing when there's a licensing. The DMC must stick to the trial until it's over unless they're told otherwise. Thank you very much for your patience and listening, and if there's time, Paul will lead us in some questions.
Thank you, Dr. Herson, for that wonderful presentation. We do have time probably to address one question, so I'll go ahead and ask that, and then you can respond to it. The question is, do you see any benefit of proper Bayesian analysis?
Yes, I think the problem that will occur here is what is proper Bayesian analysis. We don't know yet. I think a Bayesian analysis, as you know, has to be supported by lots of simulations to check frequentist properties and sensitivity to different priors. So all of this creates a lot of work and uncertainty with people who disagree with what the prior is. I do think that Bayesian analysis is useful for an individual decision, but not yet for regulatory. Now, having said that, if the DMC members are willing to rely on the statistical member and say, okay, I've chosen this prior, you can count on me, and we do this, that may very well be useful, but the physician members have to be very knowledgeable as to what is going on and how this method differs from frequentist methods.
Okay, well, thank you for that response. That is all the time we have for today's webinar, so I certainly want to thank all of our attendees. You will also be receiving a follow-up email with a copy of today's slides, as well as any responses to the questions, plus a link to Dr. Herson's book. So thanks again, and we look forward to having everyone attend our next webinar, which will be announced at a later date.